So now we're on to topic 3 about chemistry of life and biomolecules. Let's start here. So carbon is the main element of human body and makes up 40 to 50% of dry mass. And humps is also a really abundant elements in the organic molecules. Organic molecule is what makes up the backbone of organic life. Backbone is basically the most majority of primary structure components are called backbones. We get into some very basic stuff, but there is molecules, what molecule means, bond, chemical bond, form of attraction between attachments, called covalent hydrogen ionic bonds, very basic chemistry. Covalent bonds share electron, they are strong bonds that hold together. Yeah, just remember that covalent bonds are strong, maybe. And because we say hydrogen bonds are really weak in biology. There's double, triple bonds. Smaller the bond, the stronger and the more stable. Not sure if I wrote that. Yeah, so covalent bond, ionic bond, non-electrical bond. So hydrogen bonds are weak and covalent bonds are a lot stronger. And smaller the ions, atoms, stronger it is. So there's the small atoms such as CHN and O. CHNO that form these very strong covalent bonds. So there's like non-polar and polar covalent bonds, just all about if they are being shared equally or not. If there is no equal sharing, then there is polarity. So there is a partial charge on one side and another, like water has partial charge. Otherwise, like methane, CH4, they are equally distributed, so there is no polarity. Water is made by covalent bonds unequally between these two bonds. So electronegativity is what explains these non-polar polarness. Hydrogen bonds are weak, constantly formed. They are also important for holding the DNA together. So DNA together are held by nitrogen acids. Polar, hydrophilic, hydrophobic, carbon-based. Biomolecules that form cells are composed of carbon skeleton. So, where am I here? Yeah, so carbon skeleton is the backbone of all the organic molecules because carbon is what makes everything varied and diverse in organisms. So carbon is the reason that molecules are able to be diverse and organisms are able to evolve because carbon is very branched. Carbon can be very branched, that's why. So form and functions are always intrinsically linked, meaning the shape is always in parallel with the function. So different shape equals different function. That's a lot mentioned in biology. So structures are what defines one's function. So now talking about biomolecules. Biomolecules are all molecules that are made in the organism, in the living organism. So anything that human, animal, plants can make are called biomolecules. There are 3 categories of biomolecules that can form polymers, which are these 3. So there is the carbohydrate, nucleic acid, and proteins, and lipid is not included in here. Biomolecules are composed of monomers that can make macromolecules such as lipids, which is a big molecule, or polymer, which is repeating subunits of monomers, which is not the case of lipid. And polymers are, like I said, carbohydrate, nucleic, proteins. They are just repeating sequences of these monomer subunits. So dehydration and hydrolysis. Dehydration synthesis is the process of creating water creating water because there's a H and the OH group on each monomer. They're gonna be linked, and after they get linked, you can see right here. Oh no. After they get linked, they will produce the water. So the water is coming out. So this H2O is coming out. That's why it's called dehydration. But it's called a synthesis. Dehydration will make you think like it's the opposite, but no, it's formation. Synthesis because two monomers are going to be attached together. And hydrolysis is the opposite. Hydrolysis is making this into that. It's making these guys into that guy. Into smaller breaking down. So breaking down, hydrolysis. Now talking about these three, there is energy structure. Energy structure is the carbohydrate. So fuel and structure is what carbohydrate mainly do, and they also do other things like signaling and stuff. But we'll talk about that later maybe. In cell walls, the structures are there's cell wall in plants, bacteria, and algae, which are cellulose and chitin and other stuff that form cell wall. These are all polar dissolve. Not sure what that is. And nucleic acids are the encode and transmitting info that are genetic info. So nucleic acid is what forms DNA and RNA, or I should say nucleic acid. DNA and RNA are nucleic acids. Proteins, structures and catalysts. They can do chemical reactions. Enzymes are also proteins. There are a lot of things in proteins. They provide structural support and act as catalysts to facilitate chemical reaction. There's a lot of other functions, so that's a very brief explanation. And where's my lipid? Where's my lipid? Did I not talk about lipid? Okay, well, I didn't talk about lipid, but lipid is for membrane and storing energy and acting as signaling molecules. So signaling molecules act as membrane, cell membrane, composing cell membrane, store energy, signaling molecule. Just remember that. Carbohydrates, there are two roles, like I said, fuel and structure. There are different types of carbohydrate, monosaccharides, disaccharide, and polysaccharides. Monosaccharides are of 6 sugar, so hexose sugar, such as glucose, fructose, galactose. There's also mannose. Oh my god, that's so thick. And these are all isomers of C6H12O6. Those are monomers. Monomers have 6 carbons. And disaccharides are basically 2 of this. 2 of monomers will make a disaccharide instead of monosaccharide. And 2 sugar will of course be the double of that. So instead of 6 carbons, now they have 12 carbons. And these monomers, glucose, fructose, malactose, and mannose, can make up sucrose, lactose, malactose, yeah, so it's the 3 that we need to know. Sucrose is made out of glucose and fructose. So fruit and gluc will make sugar, basically. Sucrose. Lactose is made out of glucose and galactose. Glucose and galactose will make lactose. And maltose is the one made out of glucose and glucose. Glucose and glucose. Glucose and glucose will make maltose. Maltose. And just to remember that most of them are ring formed. So monosaccharides can be either ring form or of linear form, but they're mostly 97% ring and 3% linear or chain. And they always have 6 carbons instead of 5. And aldehyde group is a CHO group that is very polar and dissolves in water, and that's basically the reason that they're hydrophilic. and they form rings in aqueous solutions. They have aldehyde group. There is the D-glucose dextro and L-glucose for levo, and cannot be used by cells if it's a L-glucose. There's only the D-glucose that we're gonna concern about. And those are enantiomers. There are two enantiomers of the aldohexyl sugar. So they both have aldehyde. Yeah, and these are called aldehexyl sugar. So there's alpha sugar and beta sugar. Alpha sugar is where OH is facing down, and beta sugar is where OH is facing up. They are isomers. And alpha is for fuel, hydroxyl group going down. Beta is hydroxyl going up for structure. So there are three function of monosaccharides. These are monosaccharides, primary for energy. Typically glucose, main source of energy for cells, building block for larger carbohydrates. So fuel, making larger carbohydrate like disaccharide, polysaccharide, and making other biomolecules. So primary source of energy, and making carbon, making biomolecule, just making a lot of stuff. So monosaccharides are the subunit of lots of things. For example, amino acids. They're able to make amino acids. Monosaccharides are what makes amino acid, which makes proteins. And nucleotides are the DNA and RNA and stuff. Fatty acids are for membranes and other heat protection and stuff. Glycoprotein, glycolipids for signaling, for cell recognition, for hormones. So now let's talk about the bonds. So carbohydrates are forming bonds. Well, carbohydrates are made of these monomers of monosaccharides with glycosidic bonds. There's alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds. There's also beta bonds. We're going to talk about that later. So alpha is all about alpha sugar, of course, alpha glucose and alpha betas. Glucose. Alpha 1,4 is linear because of the 1 and 4. 1 meaning 1 carbon, 2, 3, 4, 5, 6, right? 1 and 4 will make a linear chain. 1 and 6, well, 6 is here, so if they chain together, they're going to make a branched one. And 1 and 4 is less easier to consume because they're linear. Whereas branched, if it's branched, it's easier to consume. So they are more, they're faster. They're short period. They're quick energy. Yeah, alpha 1,4 is a long-term energy, linear shaped. 1,6 is branched and is a short-lived. Well, not short-lived, but yeah. So glycosidic bonds are made between the hydroxyl group. These hydroxyl group carried by carbon 4 or carbon 6 on different monosaccharides. Yeah, so it is the carbon 1 monosaccharide. Oh no, that's illegible. Monosacharide. And the carbon 4 or 6 hydroxyl group. So monosaccharide of 1 and 4 or 6 hydroxyl group is what make these glycosidic bonds. I already talked about the ribose. And those bonds are also what makes these saccharides. So they have some functions. Their functions are of energy storage and structure support. Energy storage is in the alpha glucose, whereas structure support is in the beta glucose or beta sugar, beta glucose. The first function, storage of polysaccharide in animals and fungi, is called a glycogen. So glycogen is a very big molecule of alpha 1,4 glucose, 1,4 and 1,6, 1,4 and 1,6, and glycogen and glycosidic linkages, liver cell regulating blood, and they are in the hepatocyte cells in the liver, and muscles where there's glycogen stored. So they are long-term. I think they are long-term. Oh, it's a short term. It's a short term. They're short term, but they're what liver use to regulate blood sugar. So they are able to either hydrolyze them or form them to regulate blood sugar in the body. So that's done in the liver. Microstructures. So structure support. Now we're talking about, actually no, I still have to talk about starch. So starch is, there is what amylose and amylose and amylopectine. Amylose is 1,4, amylopectine is 1,4 and 1,6 glycosidic bonds, and amylose is linear. They are helicopolymers, long-term storage, 20 to 30% of starch, so starch are amylose. And amylopectine are the branched, 70 to 80% of starch. So most of the starch are amylopectine, 20% of starch are amylose. And they're more branched and easier to break down, so they're going to be a faster consumptions compared to the amylose because of their 1,6 glycosidic bonds, whereas alpha only have amylose only have 1,4 glycosidic bonds. Structure support. So plant use starch and animal use glycogen. Yeah, that's the difference. So starch is the amylose and amylopectine. They are the plant source of energy. So plants will use starch. Linkage of mainly alpha 1,4, but it could be 1,6 as well. So that's what amylopectine look like. That's what amylose look like. So they can be hydrolyzed to digest these alpha 1,4 glycosidic bonds by the amylase. So amylase can hydrolyze these. Hydrolyze those. Polysaccharide plants. So the betas ones are linked together, forming cellulose and structure support, linked through 1,4 glycosidic bonds. So there's no such thing as 1,6, I think. Beta is 1,4 glycosidic bonds for, like I said, the cell wall components. And these are linear and they're non-digestible. So they are fiber, lots of fiber, are beta, so they can just go through the digestive system for fluidity. So cellulose, for example, needs cellulase to digest, which is only found in bacteria, fungi, and protists. So bacteria, fungi, and protists have cellulase, which can break down cellulose. They are otherwise indigestible for animals because we don't have enzymes. Dietary fiber. They're dietary fiber. Produced by photosynthesis. Yeah, so apparently these cellulose and beta are produced by synthesis. So other function of polysaccharides are of cell recognition, such as glycoprotein and glycolipids. And dietary fiber, which are indigestible polysaccharides, and for gut health. Yeah, so they do signaling, hormones, and cell recognition, promote bowel regularity, support healthy gut. So dietary fiber for the beta ones, they're also good for healthy gut microbiota and bowel regularity and digestible. And that's it for topic 3.